throughout our lifetime, we're exposed to thousands of different infections. So different strains of cold viruses, different flu viruses, measles, chickenpox. If we get a cut, we can get different types of bacteria. And our immune system is really remarkable in pretty much no matter what infection we get, it can fight it off. And actually, every day, it doesn't know what infections we might come across, it doesn't know what viruses might be circulating even in this room. But nonetheless, we can pretty much fight things off. And this is because our immune system shows remarkable preparedness. And every single day, we generate 30 million new antibody genes that makes our immune system really prepared so that we can fight pretty much any infection that comes our way. That's great, but we don't have enough DNA in our genome to make 30 million full new antibody genes every day. So what the immune system does is it uses this really neat trick of making the bit of gene that encodes the part of the antibody that recognizes the invading virus that we see here. So we make just this bit of DNA from scratch in individual white blood cells. And we do that by joining bits of DNA together. So rather than having the gene preformed, the white blood cells take pre-existing bits of DNA and link them together in different ways. So in one cell, they link them together like this. In a different cell, you link together different bits of DNA, and so on and so forth, so that you can generate millions of different combinations, making millions of different antibody genes. And this, as I say, gives us a really effective immune system, without which we'd probably die in the first year of life from infection. But the sting in the tail is that to make these antibody genes, so to link these bits of DNA together, we actually have to break and rejoin our DNA. And that's an incredibly dangerous thing for the cell. And the story I'm going to tell you is how mistakes in this reshuffling of DNA to make antibody genes is a cause of mutations that are found in childhood leukemia. So childhood acute lymphoblastic leukemia is the most common childhood cancer, with about 650 new cases per year in children and young adults in the UK. The peak age of diagnosis, oh dear. I think the technology's just failed me there, press the wrong button. But anyway, the peak age of diagnosis is between 2 and 5, but we can get acute lymphoblastic leukemia at any age. And it's a really malignant cancer, but thankfully the cure rate for children is currently around 90%. So 9 out of 10 children will get fully cured. The problem is still for the 10% who relapse, where mortality is still too high, and that's why we are continuing to research into how we might prevent and block progression to relapse. So acute lymphoblastic leukemia develops in 2 stages. So in the first stage, we've got these big changes in chromosomes in the cell. And one example is where we've got a bit of one chromosome that's got joined onto the end of another chromosome. And these chromosome rearrangements are actually really common. And so this is work from 2 colleagues at the Institute of Cancer Research in London, Professor Sir Mel Greaves and Tony Ford. And what they did is looked in the blood spots that are taken at birth, and they saw that this actual chromosome change that I showed you on the previous slide, that's present in 1 in 100 of us. But thankfully, 1 in 100 of us don't actually get cancer. What we need is, in order to get full-blown leukemia, we need this second step, we need a second set of mutations that can lead to leukemia developing. And that happens in about 1% of the people who have this initial chromosome alteration. And errors in the production of antibody genes, so this reshuffling of DNA to make these antibody genes, errors in this process account for about 40% of these mutations. And so, as I said, to make these antibody genes, we need to reshuffle our DNA to link different bits of DNA together. And when we do this, when cells do this, what happens is these proteins called recombinases, shown here in blue circles, bring the bits of DNA together, so this orange block and this blue block, bring them together, then join them to give us the bit of antibody gene, and the bit of DNA that is between these bits of DNA, oh sorry, forgot I've got another model here. This bit of DNA, when we join these two bits of DNA together, then the bit of DNA in between is kicked out of the genome, so this bit of DNA here is kicked out of the genome and is circularized to form what we call this ESC, size signal circle. So we kick this bit of DNA out, and this is just hanging around in cells as a byproduct. The way in which errors in this process can lead to leukemia are twofold. Firstly, the recombinase, when it's bringing these bits of DNA together, recognizes specific sequences in the DNA that are shown here as triangles. As well as these sequences being found in the regions we want them to make these antibody genes, there's also thousands of copies on different chromosomes. And what happens is the recombinase, rather than making the antibody genes and putting together these blocks of DNA, it actually changes DNA on other chromosomes completely independently. The second way in which errors in producing antibody genes can lead to leukemia is something we published, oh dear, I keep going to change slides there, but I'm going to do it here. Is a mechanism which we discovered and published in 2019. And it's this byproduct, this circular DNA that's kicked out of the genome. It's got these triangle sequences on it. It binds to the recombinase, causes a break, so it cuts, releases the broken DNA, and then runs to cut elsewhere in the genome. So the recombinase binds this byproduct and can go around the genome generating breaks. And we found that these breaks correspond to the breaks that are found in childhood leukemia. So which mechanism is happening most? Is it just the recombinase mutating, seeing the triangle bits and mutating the genome? Or is it this cut and run? And we wanted to understand that in order to say, well, can we actually block the reaction? Can we stop the reactions that might lead the cancers to go from just being a cancer that can be cured to one that relapses and has too many and gets too many mutations? So we looked, going in the wrong place again, we looked in the genomes of, so we had the whole genome sequences of 200 acute lymphoblastic leukemia patients. And as had been shown before, we found that about 40% of the mutations in those patients were found by mistakes in the production of antibody genes. But of those 40%, about 3/4 of the mutations are caused by cut and run, and only 1/4 by the recombinase binding to the triangular bits elsewhere in the genome. We then said, well, okay, what's actually causing the mutations that lead to relapse? Because if we can stop that, we can maybe find ways of stopping disease progression to relapse. And so when we looked there, what we found is, and so this is just the different genes that are often mutated at relapse. And what we found is that there's significantly more of them, a lot more of them, are mutated by cut and run rather than the other mechanisms. So that's saying, so we thought this could be important because it's saying that cut and run was leading to more of the mutations, and it's also mutating the relapse genes. And we think there might be a way of blocking cut and run because the way in which the recombinase binds these circly bits is different from how the recombinase binds to produce antibody genes. So what we're currently doing is looking in detail at how the recombinase is binding to the circular byproduct to find ways in which we can block the recombinase binding, to try to block this cut and run reaction, to find ways of blocking disease progression to relapse. The other thing we thought was, well, okay, this circly bit is needed for cut and run. And this cut and run reaction is causing the mutations that occur when patients relapse. 10% of patients relapse, so are there maybe higher levels of these circly bits in the patients who relapse? So we got samples that had all been taken at diagnosis from Vivo Biobank. So we got patient samples and looked to see what are the levels of these circle bits at diagnosis in patients who relapse and in those who don't. So the patients who relapse are shown here on the left, the patients who don't are shown here on the right, and what the dots are just the levels of the individual different circly bits in each of the patients. And these are all different patients along the bottom. And what you can see is we've actually got much higher levels of this byproduct bit of DNA in patients who relapse versus those who don't. So we thought, if we've got these higher levels, can we use this to pick out the patients who might relapse early? So currently, what clinicians do is they treat the patients using their best treatments for the particular leukemia subtypes, and then look after a month to see how much disease is left, and then judge from that whether or not the patient may go on to relapse. These samples were all taken at diagnosis. So if we can look at the levels of these circles at diagnosis, then we can maybe pick out the patients who might relapse much, 4 weeks, 4 or 5 weeks earlier. So we tried just looking at the levels alone of a library of these circular DNAs, and that alone wasn't enough. But what we also discovered is that this circle DNA becomes processed a lot more in the patients who relapse. So without going into too much detail, when we form this circle, that I keep playing with this stickle brick here, but when we form this circle, we put these two triangular bits absolutely up against each other, and it forms a specific sequence in DNA that this protein recognizes. But when we get the processing that we've discovered, then the protein can't recognize it. This processing happens much more in the patients who relapse. So what we're doing is we developed an assay where if we've got this high level of processing plus the high levels of the ESC, we thought that could be a good indicator of a high risk of relapse. But if we've not got the processing and we've not got high levels, then it's a low risk of relapse. And so we've looked in the samples from Vivo Biobank again to see, can we actually pick out the patients who might relapse? And across all the different subtypes of ALL, we can, but we've got a couple of false positives, which actually is no good in the clinic. But when we look in the two most common types of acute lymphoblastic leukemia, then we are able to pick out a reasonable number of the patients who will relapse just in the diagnosis samples. So what we're doing now is speaking with the diagnostic lab over at St. James's to see if we can actually expand this up to look in more patient samples to be able to see, actually, can we develop this as an assay to be able to pick out the patients who will relapse early so that clinicians are informed to be able to decide the best treatments for those patients much earlier in the treatment cycle. So what I've been describing to you is that we've been looking at how errors in production of antibody genes lead to leukemia. We found that this cut and run mechanism seems to be prevalent in mutating the genes that are linked to relapse and in causing the mutations generally. And so what we're now working on is finding ways to block this reaction, to try to find ways of blocking disease progression to relapse. And we've also found this new marker that of these bits of circles that when it's present at high levels at diagnosis seems to be a marker of relapse. And so we're trying to develop a new assay to be able to pick out the patients who might relapse early. And finally, I'd like to thank the people in the lab who've done the work, our collaborators, and most importantly, I'd like to thank the funders, because none of this would be possible without funding. We're about to start our fourth grant that's been administered by CCLG, with funding generously raised by the Little Princess Trust and the Harley Staples Cancer Trust. And I'd also like to really thank Vivo Biobank, because without them having the faith in us to give us all of those patient samples and people who've allowed their samples to be used by Vivo Biobank, then we just wouldn't be able to do this work. And thank you for listening.